Hello, my name is Kaleigh, and today I will be discussing my research from the past semester, investigating the effects of expressing chaperone 60 as a transgene in model tobacco through Agrobacterium mediated transformation. Plants experience multiple types of abiotic stressors, including heat, salinity, drought, and more. During times of heat stress, plants utilize chaperones as a means to create abiotic stress resilience. Chaperones specifically aid as a catalyst in the folding and refolding of proteins to keep proteins from degrading and maintaining their intended function. Introducing a heat shock protein such as chaperone 60 into plants would improve plant tolerance to increasing temperatures. Chaperone 60 is a heat shock protein commonly found in Pyrococcus furiosus. Pyrococcus furiosus functions at an optimal temperature of 100 degrees Celsius. Incorporating the chaperone 60 gene from such an extremophile will hopefully lead to heat resistance with plants. It is also expected that when our gene is active, there will be protein and membrane integrity, and it will not undergo degradation due to the high heat. Last semester, an HVHSP promoter was used in the construct of chaperone 60. However, when the plants underwent heat treatment, there was no observable differences between the transgenic tobacco with the wild-type tobacco. This can be attributed to the fact that the HVHSP promoter is a promoter that is only active under a certain range of temperatures. Therefore, a new construct has been created utilizing a cauliflower mosaic 35S promoter. The 35S promoter is constitutively active, meaning that it will always be on and does not require a certain temperature. This research aims to express the chaperone 60 gene through Agrobacterium mediated transformation in a tobacco model and compare heat resilience of the transgenic tobacco to its wild type. Once the two constructs were created and were successful, we then utilize Agrobacterium mediated transformation of tobacco to insert our gene and expression cassette into the plant genome of our tobacco. This was done by saturating leaf pieces in a solution containing our gene as well as other nutrients. The leaf pieces were then transferred to shoot induction media, and after a few weeks were then transferred to rooting media, and then were transferred into soil. The putatively transgenic plants then underwent heat treatment in order to see if the chaperone 60 gene had truly been expressed. As a result, the transgenic seedlings were exposed to the high temperature of 45 degrees Celsius, and after heat treatment, there is no phenotypically observable differences between the transgenic tobacco and the wild type under control of the HVHSP promoter. We have just recently generated a construct under the control of the cauliflower mosaic 35S promoter, which will be introduced into tobacco and expected to increase heat tolerance. This means our future work will be composed of the same steps of undergoing Agrobacterium mediated transformation, planting into selection media, then planting into rooting media, and finally the soil. We hope to see observable phenotypic differences in response to the heat treatment under the new promoter, which includes less wilting and more plant integrity. If there are any questions, please feel free to reach out to me, and I would like to give a quick thank you to the UNC Greensboro Biology Undergraduate Research Award for making this research possible. Thank you!